Richard Rosen. I am a vitreoretinal surgeon at the New York Eye and Ear Infirmary of Mount Sinai in New York, New York. I'm involved with a number of studies. In our laboratory, we work with high-resolution imaging, adaptive optics, OCT angiography, and most recently, what I'm talking to you today about is metabolic imaging using flavoprotein autofluorescence. Flavoprotein fluorescence shows oxidative stress in mitochondria in a way we've never been able to measure it before in living patients, and it's an indication of the disease which is beginning to occur for a number of reasons. It can be due to pressure, it can be due to increased blood sugar, it can be due to aging, and the interesting thing about this technology is it's the first technology that measures metabolic change that actually shows reversibility with some of our new treatments. So what I presented at the subspecialty day yesterday were a sort of collection of studies that have gone on between our institution, the group at the University of Michigan, the group at Stanford, and the group at Cleveland Clinic at the Cole Eye Center, looking at how flavoprotein fluorescence may become a clinical tool for monitoring diabetic retinopathy, age-related macular degeneration, and glaucoma. The technology is fairly simple. The patient sits in front of the camera. A small blue light stimulates the macula or the optic nerve, and then the camera detects the green fluorescence and measures that fluorescence, and that is the indication of where the macula in general, it measures an area of about 20 microns in distance.
So are you talking about fluorescents that are injected into the eye or biofluorescence that naturally occur in the eye?
This is the natural fluorescence that's produced by the mitochondria in the cells when they're under stress.
Please tell me about the ongoing ARID study and the effects of oxidative stress.
So there are a number of studies. One that was done about a year ago by Robert Rich and his research group looked at a combination of neuroprotective components for glaucoma, and they mixed this and they did a placebo-controlled trial where they gave two groups of subjects either the real thing or the placebo for a month, and they showed significant reduction in oxidative stress in the group that was treated with the antioxidant mixture. They used a combination of curcumin and ginkgo and a large variety of nutraceuticals that we've known have some benefit, but he put them into a proprietary mixture. More recently, we're starting to look at just the impact in the ARIDs, and in a short study that was just done, and it's not completed yet, at 1 week we noticed that there was a drop in the oxidative stress in the subjects who were taking the ARIDs component. We've also seen that, most interestingly, in patients who were being injected for diabetic macular edema, that while at 1 month they saw an improvement in their visual acuity, it really wasn't, we didn't see that much of an effect from the OCT thickness, but in fact the flavoprotein fluorescence mirrored the visual acuity response much better than the OCT. It was much more sensitive, and yet it's objective as opposed to the subjective response to the vision.
How long will these studies be going on?
Well, there are a number of them that are ongoing now. There are a number of different nutraceuticals and some pharmaceutical agents that are being tested, and they're at different stages of development. So there's some working in glaucoma, there's some looking at age-related macular degeneration and diabetes. So the results will be coming out, and then they'll be published, so they'll be available. The current technology, though, is in a commercial package. It's been, it was a proprietary technology which was originally in the laboratory, and now it's in the commercial Icahn camera, which is fairly easy to use, takes less than a minute to do the test, and has very good repeatability of less than 4% between sessions.
Is this imaging technology available in the market today?
Yes. Of course, the one that is being sold is not the one, it won't be modifiable, but it will be basically operate the same way.
Where can someone follow your research or get involved in one of these clinical trials?
Well, you can find me through the website at the New York Eye and Ear Infirmary. I can tell you about the studies that we're involved in. The company that actually has the technology is called Oxidative. They're out of Ann Arbor, and they have a website, so you can contact them directly too. They'll be able to tell you where the various trials are ongoing, either in this area at Stanford or in Cleveland area. And the plans are that they will expand the studies to a number of academic centers throughout the country.
As always, Dr. Rosen, thank you for taking time to talk with us.